This is what this slide may look like if you had an inherited retinal disease. Hello, my name is Nicholas Ponticus. I'm a postdoc at the University of Ophthalmology and a data scientist at Morpheus Eye Hospital. I specialize in analysis of inherited retinal diseases, both from the genetic perspective and from the image analysis perspective. My proposal, NIHR, is to develop a web tool to assist the genetic diagnosis of inherited retinal diseases using machine learning. Inherited retinal diseases are estimated to affect around 25,000 people in the UK. They are the leading cause of visual impairment in the working age population, and they are typically single gene disorders, meaning that mutations in a single gene are sufficient to cause the disease. Around 200 genes have been identified so far. There's ongoing gene therapy trials, which are targeting around 10 of these genes. There's around 36 clinical trials at the moment, which are recruiting patients. Of the 25,000 estimated IRD patients, it's estimated that 60% of them have a genetic diagnosis. Currently, the genetic diagnosis process involves referrals from optometrists, of which there are around 14,000 in the UK. And these referrals, it's worthy to mention, are mostly false positives when it comes to inherited retinal diseases, because these are rare diseases and they are difficult to spot if you're not a specialist. These referrals go to specialist eye hospitals. Patients are seen and examined by ophthalmologists. And if the patient is thought to have an inherited retinal disease, then DNA is taken and sent for sequencing and can be analyzed in one of the 13 genomic medicine centers. This whole process can take a while of finding a genetic diagnosis, and there's many patients that I've spoken to who've been waiting for more than 5 years to have a genetic diagnosis. So my goal is, through the development of this website, to lower the number of false positive referrals by using it as a screening tool, and to also diminish the time till genetic diagnosis by facilitating the integration and prediction from data. I'm hoping this will lead to a higher genetic diagnosis rate and hence more patients eligible for clinical trials. My objectives of NIHR are to emulate the diagnosis process with machine learning. So this involves genetic diagnosis from retinal images as a first objective, genetic diagnosis from clinical symptoms, which are what a doctor would report from clinic, and finally identification of the cause of mutation by using the DNA sequencing data and combining it with the clinical and genetic data. Here's an overview of what the plan is going to look like. So in the first year, I'll be gathering the 28,000 images that we have at Morpheus. These are images which are labeled with genes, and we're going to be feeding these to a convolutional neural network, which is a machine learning tool which is able to do image classification. The output of this network is gene probabilities. So the way to interpret this is how likely that image is to be of a patient with a mutation in that gene. The second objective is the integration of the clinical data. So this involves text mining of records, and it also involves analysis of images and extracting clinical symptoms from images. So, for instance, from this text record and from this image, I would extract from the image rod cone dystrophy or abnormality of the orbital region, and from the text record I would extract things like mild hearing impairment, autosomal recessive inheritance. These terms are going to be standardized using the human phenotype ontology, and then we're going to feed them to a machine learning approach called a random forest to again obtain gene probabilities. Finally, I'm going to take the genetic data from the 9,000 patients we have, feed it for our genomics pipeline, and then use the HPO terms from the previous step combined into the Phenopolis platform, which is a web tool I developed last year, which is able to prioritize genes based on genetic and HPO terms. And again, we will be obtaining gene probabilities from this. Finally, we will combine the gene probabilities from the different approaches in order to obtain a final consensus on what the likely causal gene is for this particular patient. Here's a screenshot of the website. It's I would say, a demo version of the website where users are currently able to upload images and obtain a prediction. So the result is going to be an efficient genetic diagnosis tool for IRDs. It will be efficient because it will limit the number of false positive referrals. It will make the process of genetic diagnosis easier by integrating imaging and genetic data, processes which at the moment require different specialists to look at in parallel. It's going to be the world's largest data set of IRD retinal images and symptoms through the Moorfields data and users' contributions. And this is important because machine learning needs large amounts of data in order to be efficient. It's going to be a portal for doctors and patients to communicate, because it will be the intention is for both of them to be users of the system and to be able to communicate and share data. The data will be owned by the patient. The patient will be able to access his data and download it, and will also be updated as to what's happening in his diagnosis to stay informed. And the platform will also be able to preemptively predict IRDs. So my training and development plan is going to be to attend courses in my first year in clinical ophthalmology, text mining of clinical records, and image analysis. And I've arranged strategic research visits at different sites where I'll be either having data sharing agreements or exchange of knowledge and skills. And I'll also be attending conferences every year where I'll be presenting my work and engaging with patients. Finally, I'll leave you with some comments I've had, some feedback I've had from patients already, some of the positive feedback I've had from patients already about this project. Thank you very much.